So we did prophase 1, where we saw crossing over, metaphase 1, where we saw independent assortment, and then in anaphase 1, we're going to see the separation of the homologous chromosomes. So our homochromos are separating. Remember, that's very different than what we see in mitosis. Okay, and at this phase of anaphase 1, dun, da, da, dun, da, 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 our cells are now haploid because our chromosomes are now one set of chromosomes. So what we see in telophase 1, we still see cytokinesis happening at the same time. We're seeing these nuclear envelopes reforming. We're seeing the DNA relax from Cheeto form back into spaghetti form. And now we have 2 haploid cells, but we're not done yet, okay? Because our DNA is still paired up in the sister chromatids. We need to get those separate, okay? So sometimes what we see is that these cells, these haploid daughter cells, are going to go back to interphase, but sometimes they're going to immediately go through the second meiosis process. We see interphase between meiosis 1 and meiosis 2. And that does not include the S phase. So this phase where we're synthesizing the DNA, that gets shut down, right? The whole goal of meiosis is to take our genetic material and split it in half. If we duplicate it halfway through, then we're defeating the purpose of the whole thing in the first place. As we go into meiosis 2, this is going to look very much like mitosis did. So in prophase 2, what we're going to do is undo what we just did, right? So we're going to get rid of that nuclear envelope. We're going to have spindle fibers reform. We're going to condense our DNA from chromatin into chromosomes. Metaphase 2, we're going to line our chromosomes up along the equatorial plane, and we're going to have the spindle fibers attached to our sister chromatids. In anaphase 2, those sister chromatids are going to separate. In telophase 2, we're going to see the nuclear envelopes reform. We're going to have cytokinesis, and we're going to end up with 4 genetically distinct cells.